Welcome to today's lesson. We're going to have a review of DNA databanks, and these are electronic collection of DNA profiles of individuals, and they are not necessarily related to health, that is diseases or disorders. So they are usually profiles that are organized in different indexes for the purposes of forensic studies, for example, population DNA data bank, criminal forensic data banks, wildlife data banks. And it is good to note that they differ from nucleotide, which are now DNA, RNA, as well as protein databases that are connected to health. So here we look at what are these nucleotide databases so that you can differentiate them from the DNA data banks that we are using for forensic purposes. The nucleotide databases are a collection of sequences, full or partial, that usually will be stored in places such as gene bank, reference sequence banks, TPA, protein data banks, and they consist of genomes, like the whole of the genome of human beings, the genome of plants, genes, the various genes that are responsible for production of functional products within the system, and then transcripts such as mRNA and other RNA, sequence data, and usually they provide foundation for biomedical research and discovery. Now this, for example, we have said gene banks, data bank, DNA data bank of Japan, TIGR, yeast and microbe databases. We also have specialized databases that will have things like ESTs or express sequence tags, STRs, etc., also stored in places such as NCBI and TIGR. So these sequences of genomes, transcripts, proteins, and also specialized tags like express sequence tags are connected to general sequencing outcomes. So during biomedical research, we have sequencing technologies or sequencing of genomes, partially or full, like the Human Genome Project. So where do they store these sequencing projects? We store them in nucleotide databases. We could also be having genetic defects databases specifically from the study of genetic disorders, as well as certain disease profiles. So let us look at now the various DNA data banks that are important, especially in forensic studies. One of them is a population DNA data bank, and this one is a database consisting of DNA profiles of anonymous people. And anonymous to mean people have been involved in research, randomly involved in research, so that we can have epidemiological data of certain populations within the world. So it is anonymous, and then we obtain their DNA, and this is required for calculation of the following parameters when it comes to forensic studies. That is, for example, probability of a match. How are we able to match individuals so that we can identify people? How are we able to match a father to the possible children they have sired? That's paternity testing. How are we able to calculate relationship indices such as sibling test or any other relationship like mother and children, etc. So usually these calculations give us what we call power or weight of evidence. And I want to define a few terms such as probability, that is the frequency of the occurrence of an event. And this frequency, usually we will define it, or we will generate what we call probative value. And a higher value is obtained if the evidence can prove something in the court. we want it to give us high value. The probative value should be of high value so that the evidence is able to prove something. So usually the lower the probability of an event, that is, the lower its frequency, okay, the higher the probative value. So we need a high probative value, and this is indicated when we have the lower probability of an event occurring. That means if you're identifying people and we are using a certain marker, we want that marker to have a lower probability of being available, too available. Because if it is too available, we cannot narrow down to specific people. Then we have what we call power of discrimination as an important term in the calculation and statistical analysis of DNA evidence, or what we call random match probability. And this is now where we are looking at individuals by chance alone. We want to see how well is the loci that we select able to individualize. Okay, it's able to individualize that once you have selected two people randomly from a population, you're going to get identical phenotypes or all genotypes by chance alone, so that it can give us a high power to discriminate between individuals. We have what we call PD, power of exclusion. This is how powerful our marker panel, because we usually will use a panel using various markers like we can see here in excluding particular genotypes. For example, in this case, we have these genotypes of African American, Bahamian, Jamaican, Trinidad, Caucasian, and Hispanic populations using this marker profile. So we want to identify the power of exclusion, okay, of excluding particular genotypes once you're comparing even different populations across the world, so that at the end of the day we are able to say that the evidence that we are having, we are sure this is an African American, or this is a Jamaican, or this is a Caucasian individual, among other calculations of evidence in the court of law. So in this case, usually what we will develop, one of the main population databases is what we call an allele frequency database. We talked about alleles, the different forms of genes, and usually this you get at least a minimum of 100 people, and the frequency differs from population to population. So every country develops their allele frequency database. Like in this case, there is an allele frequency database, genetic data on 10 autosomal SDR, okay, loci in this Bangladeshi population. So we will need to have different populations being studied for several markers, all of them or partially. How do we use these statistics to determine the weight or the power, or how good our data is at being able to discriminate between suspects and people who have actually been involved in a crime? So usually there will be development of what we call reference profiles using population data banks, so that now this is the basis of us comparing and giving us the weight. So once you're able to get all the loci, like you see, this is a full profile of that 13 STR markers that we have studied in DNA fingerprinting. In this population that was here, you see they did 10, okay? These ones did 10 of the marker, the STR markers. So in this population where we have identified a full reference profile of markers, we can see that the statistical conclusion is that for you to find this profile in a population, it will be 1 in 665 trillion people. okay, to get this exact. Sometimes we also do use partial profiles, okay, so that now we have maybe loci 1 to 5 giving us a calculation or weight of 1 in 171,000 individuals. So what will happen is that a match observed at all loci that may be compared. So now using this reference, we can be able to compare once you have your evidence and see how is your evidence comparing to the reference profile. So the reference sample is still a match, just not as much information is available from the comparison, from the evidence for comparison. Okay, so you can see that if this was your evidence and we have 1 to 5 and they are matching, you can see they're exactly matching. So we are saying that it is good to conclude that this is the person. So the reference will have indicated it's a certain person. So it will be good to conclude, although it would be best if the evidence can also analyze the rest of the loci for us to be completely sure that the calculated evidence is powerful for us to make a conclusion on the crime. Then we have what we call criminal forensic DNA databanks. And now these ones are specifically forensic purpose databases that are centralized DNA databases that store DNA profiles of individuals and enable searching and comparing of DNA samples that now we collect from the crime scene against these stored samples. So these ones they have been collected in previous crimes or previous forensic situations, and now we use them to now do a comparison and search against now present crime scene DNA that we keep obtaining over time. The purpose of these forensic DNA databases was to develop investigative leads, to at least have a place to start. Before you go to the population database, can you be able to start from previous crimes or previous present forensic data that is there, that you're able to solve crime and identify individuals. In around May of 2013, China and the United States have maintained the two largest forensic databases that have contained more than 20 million for the China database and 12 million profiles for the US databases. And with time, the Chinese one has developed 410,000 hits, meaning 410,000 crime scene samples compared against the 20 million stored forensic DNA profiles have provided a match. And for the US database, about 185,000. Okay, so you can see how important it is, because now the 410 cases have already been now started and they have a direction in which they are leading to, because it's not easy to just study DNA because you are not able to like obtain DNA for every individual within the planet. So once you have a place where we have reference or stored, it becomes a place of helping us develop an investigative lead. How do you develop these ones? Usually they are developed in the process of forensic DNA analysis, so that now they are stored for future comparison and such. Because in DNA we do what we call collection of the sample from the crime scene, from the suspect, and of course also storage. Then we extract DNA. Then after we have extracted DNA, we quantify how much of DNA do we have in nanograms. And then it's important to amplify using polymerase chain reaction, because most of the DNA obtained is minute or is in low quantities. So we would want to amplify various segments. And that's why we do multiplexing, so that you are amplifying different markers or multiple markers. Then do STR typing. And we talked about STR, that's a short tandem repeats. And once you have done this STR typing, you do interpretation. For example, for this one, you see it's a male, X and Y, and now these are all the other markers that have been studied for this. You can use multiple markers up to the 13th loci. Then you interpret the results. Once you have interpreted, then you put this information into a DNA database. Okay, so that once you start searching, okay, you search so that you can identify if you're able to get any hits. So you compare the DNA profile that you have obtained to either a population allele frequency profile or now the criminal database profile, so that now you can be able to check for probability of random match to an unrelated individual. And now once you have done that, now of course this information that you obtained here will now form the future reference for now other samples that come after that. So these profiles of criminals or criminal forensic databases usually are of three types, or they include three profiles. One of them is what we call from convicted offenders and suspects. Okay, convicted offenders and suspects that have been arrested. So these are known. They are convicted or they have already been arrested. So they are known sources. We also have unknown sources, that you have collected DNA from crime scene and it cannot be connected yet to any suspect or any convict. So it is unknown. And then we have profiles from missing persons and their relatives, or you have unidentified human remains in certain criminal investigations. So you include them in the profile. So typically we are saying that once you have had the three profiles, this is how you do a typical search. You have your unknown forensic profile that you have obtained, search against the convicted offender, arrestee profiles, or among the unknown, or among the missing or unidentified individuals, and then determine if there is any association, what we call a hit or a match. And from there, you get investigative leads, positive or negative, to be able to proceed with your investigation. Now, in the US, we have what we call CODIS, or a combined DNA index system, that supports all the criminal justice in the FBI program for criminal investigation. So here, all the states, all the federal states, and other local laboratories that do forensic work will submit the DNA profiles that they obtained in the course of their search to this centralized system called the CODIS, and you'll use the 13 loci. So various profiles keep being added, keep being added, keep being added. So it becomes a centralized place for the national databases, local forensic laboratories to submit new profiles that they obtained in the course of their investigation. So there will be indexes for what you call forensic index that contains just crime scene evidence, and also there is the offender index, which now will consist of people who have already been convicted, sexual offenses and violent crimes, and today we are also including other crimes or felonies. Forensic DNA testing looks at a set of markers. I have already mentioned this, and we have the typical STR or short tandem repeat testing that looks at a few markers on on the 24 places, but at this point we have identified about 13 CODIS. loci that are being used. We also now have 23 markers on what we call the Y chromosome or Y short tandem repeats that has about 23 markers, and this one helps us in identification of paternal lineage or male samples that have been collected from crime scenes. We have another international database called iFamilia or International Criminal Police Organization that usually identifies missing persons globally through family DNA matching. So this one usually is a global database that identifies missing persons based on an international DNA kinship matching. So people or individuals and organizations across the world will contribute DNA to this database so that in the eventuality of missing people and identified human remains, we have, comparison using DNA samples from family members so that family members will contribute so that we can see if the people that are missing or identified kinship can be connected to their families across the world. Wildlife Data Bank is now the last form of DNA data banks that are useful in forensics because today we have a lot of wildlife crimes, and also there is the threat to biodiversity due to overexploitation, climate change that are causing decline in mammals, birds, fish, amphibians, reptiles since the 1970s. So wildlife data banks are useful because they are a tool for the management or stocking and helping us to understand how do we manage and reestablish wildlife across societies, and also very key in terms of conservation of biodiversity, and then also recovering what are the threatened species within the population. And then of critical to us is what we call wildlife forensics because of the criminal acts of poaching and killing of wildlife. We need to maintain databases of species ID, what are the individual identifications, and of course even the population identification of all the various wildlife that we have across our parks and our natural reserves. So this database is just like the human databases usually will contain the DNAs or DNA profiles of wildlife so that it forms a basis of us comparing or searching against the reference that is already now available with those that we find, the DNA that we find at various criminal scenes that involve wildlife. That is it on the snippet of DNA data banks.